Hello, everyone, and welcome to today's broadcast, 3D Cell Cultures and Tissue Clearing, a high-content confocal platform for the complete 3D characterization of advanced cell models, presented by Dr. Michael Johnson, Chief Executive Officer, Visikol Incorporated. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Johnson. I will now turn the presentation over to him.
So my name is Dr. Michael Johnson. I am the CEO and co-founder of Visikol, and our company is focused on 3D cell culture models, tissue imaging, and tissue analysis. Before I dive into the presentation today on 3D cell culture models and their characterization, I want to talk a little bit about our company and what it's all about. Our company is a contract research organization that's focused on transforming tissues into actionable insights. Our team is comprised of experts in advanced optical imaging, 3D cell culture models, automation, and digital pathology. We currently offer services in 3 different service areas in the spaces of in vitro screening, so working with 3D cell culture models, whole mount tissue analysis, so looking at tissues like whole mouse brains and whole tissue sections in 3D. And lastly, digital pathology, so turning traditional two-dimensional slides into quantitative insights for researchers. Through this presentation, I was going to focus on 3D cell culturing, so talk about where 3D cell culturing came from, background considerations for using 3D cell culture models and assays and their characterization. I was going to talk about high content screening, so how we actually extract data from 3D cell culture models. What are some of the current limitations of high content screening today? And talk about 3 specific case studies in which we've used 3D cell culture models in assays and services at Visikol for our clients. Now, the most basic question is why 3D cell culture models? There are many researchers who've used 2D cell culture models for years, and the 3D cell culture model space is relatively new, and the question is always asked, why are we transitioning to 3D cell culture models? The rationale for this, the rationale for so much research in this area, is that traditionally there's a poor translational gap between in vitro and in vivo. A lot of the results that we see in vitro don't actually translate to what's seen in vivo, and this results in several major problems: reduced in vitro selectivity, expanded in vivo studies, later stage failures where it's very expensive to fail, and unnecessary screening of compounds that should have passed on to later stage studies. And lastly, increased development costs and also development time to get a new therapeutic to market. The rationale for using 3D cell culture models is simply that biology does not occur within the body in 2D. Traditional 2D cell culture models are great for many applications. They're very high throughput, but biology does not occur in 2D. We have gradients, we have all sorts of diffusion gradients throughout tissues that are not replicated very well in 2-dimensional cell culture models. So the shift to 3D is having an environment that more accurately mimics the complex microenvironment within tissues and ultimately allows us to translate results from 3D in vitro studies to in vivo studies more accurately. Now, before I dive into this in a lot more detail, I just want to talk about the nomenclature. So in publications and in marketing materials, we hear these words: spheroids, microtissues, organoids, and really what they all are 3D cell culture models. I think a lot of researchers find these terms to be quite misleading, as we don't actually have little tiny organs in a dish. We don't have little tiny tissues in a dish. We're actually very far from there. And essentially we have our 3D cell culture models. We don't have microorgans just yet. Hopefully one day we'll get there, but we're not quite there yet. But broadly speaking, spheroids, microtissues, and organoids are all referring to the same thing, which are a single or multiple cell types within a three-dimensional construct that's usually between 100 and 500 microns in diameter. That can be grown a wide array of different ways, which I'll talk about in a moment here. Now, some of the common techniques for generating 3D cell culture models are using a matrix-based approach, so Matrigel, which is very popular, very common, ultra-low attachment plates from Corning, N3D magnetic printing. We're actually using magnetic nanoshuttles to cause 3D aggregation of tissues. And lastly, hanging drops, which is another very common approach for generating 3D cell culture models. Now, each one of these different techniques will really depend on what type of cells you're trying to grow, what your application is, and which one of these is best for your research will really depend upon what type of question you're trying to answer and the cost, throughput, and ease of use that you're looking for in a particular assay. There's not one technique that's going to be best for all applications. Something to really stress and to really drive home here is the point that 3D cell culture models are not a replacement for 2D cell culture models. I think a lot of researchers are trying to push the idea that these are little tiny organs and we're replacing all in vitro studies that have come before. But that is just not, frankly, true. 3D cell culture models have their place, but it's not as a replacement for 2D cell culture models for all applications. When we're thinking about designing an assay, we're looking at a couple different things, three primary factors. We're looking at cost, relevancy, so how well are we mimicking what's going on within the body, and lastly, throughput. Simply put here, we're starting with cell-based assays typically, so we're using assays that are very high throughput. They have a very low cost on a per data basis, and the relevancy also is kind of quite low. And as we proceed from having lots and lots of compounds to fewer and fewer and fewer compounds, we're moving from cell-based assays to 2D cell culture assays to 3D cell culture to animal models to ultimately clinical testing. And along the way, what we're doing is we're improving the relevancy, but we're also increasing the cost, and along the way, we're reducing the throughput. So 3D cell culture models are great. They allow us to improve our relevancy, but consequently, they're increasing the cost and reducing the throughput compared to traditional 2D cell culture models. So they have their place, but it's not as a complete replacement of 2D cell culture models. Now, there's some strengths and also some weaknesses beyond 3D cell culture models that must be kept in mind when designing an assay to use them. So 3D cell culture models, they're more physiologically relevant than 2D. That can matter for an application, it might not. So 3D cell culture models, again, are not best for all applications. It depends on your specific type of research question. It depends on your validation criteria and the type of cells and the type of question that you're looking for. So we have heterogeneous cell populations in 3D. We have a customizable environment. We have better gradients. We're more accurately mimic the environment within in vivo tissues compared to what we do in a traditional 2D cell culture model. But 3D cell culture models, it's a very new space. There's not a lot of consensus around which models are best for which applications. There's a lot of challenges with labeling, with imaging, characterization, which I'll talk about here in a moment. And currently, there's not a general consensus on which types of cell models to use for specific assays, or what types of data that should be acquired, or what kind of validation requirements are needed for specific types of models and questions. So the field is very new, and also ultimately 3D cell culture models will always cost more than 2D cell culture models, and likely always have a lower throughput than a 2D cell culture model. Now, what I'm going to dive into is how do we actually interrogate 3D cell culture models today to extract actual insights from the models? Because ultimately what we're trying to do is we're trying to better mimic what's going on within the body. And by doing that, we want to extract data from those 3D cell culture models. And there's a wide range of ways that we can do it. Again, this is a balance between cost, throughput, and some other limitations of the assay itself. So the simplest approach to interrogating a 3D cell culture model is to use a dissolution-based assay. So we're just taking the 3D cell culture model, we're distilling it down to perhaps one single endpoint, such as total ATP, and we're getting one data point from a well. This is very high throughput. The cost is quite low, but in the process, we're losing all the spatial data that actually makes these 3D cell culture models more indicative of what's going on within the body. We can also use chemical analysis, which again is going to get a single data point or a few single data points from the tissue. Again, high throughput, low cost. We can use wide field imaging. Throughput here is going to be relatively high. But the problem with wide field imaging and the problem with imaging 3D cell culture models in general, which I'll talk about on the next slide, is that if you're imaging a 3D cell culture model, it is not transparent. So if you're taking a singular picture of one of these models, essentially you're only taking a picture of the outside of the model, and you're only taking that picture on one side of the model. It'd be as if you were taking a picture of the moon. You're only really imaging the exterior of one half of the moon, and because the moon is not transparent, just like a 3D cell culture model, you're only characterizing the periphery. You're not actually characterizing the entire population of cells, which is very problematic because the entire 3D cell culture model is not one homogeneous structure. The different cells, the different components and parts of the 3D cell culture model will vary greatly in how they react to a compound or to an antibody, for instance. We can also do histopathology, so we can pull out the 3D cell culture models. We can slice them and do traditional H&E or IHC. The problem here is that this is very low throughput. It's very tedious. It's time consuming. Imagine trying to slice a 500 micron diameter 3D cell culture model and acquire a 10 micron slice or a 100 micron slice. It's very challenging to do, and it's very low throughput. And then lastly, we have confocal microscopy, which I'll talk a lot about in this presentation, which can be used to actually extract all the information from a 3D cell culture model if we use tissue clearing. So if we use confocal microscopy, just like wide field imaging today, we're going to have the same problem, which is a 3D cell culture model is not transparent. It has a lot of opacity, and because of that, we would only characterize the periphery of these models and not actually characterize the entire population. So confocal can allow you to characterize all the cells, but it requires tissue clearing to do so. And confocal imaging, because we're acquiring stacks of images from one model instead of one singular image like wide field imaging, it's going to be much, much slower, it's going to be more expensive, but we're acquiring a higher data density for each individual 3D cell culture model. Now, to expand on that point and comparing wide field to confocal microscopy, what I'm showing here is us using a tissue clearing technique we developed called Visikol HISTO-M. It's a rapid plate-compatible tissue clearing technique that renders tissues transparent through the non-destructive process of refractive index matching. So this can be done in a well plate. It's reversible. It can be done with an automated pipetting robot. But essentially, we're removing the water from the cytosol, and we're matching the refractive index throughout entire 3D cell culture models, which allows light to pass through unobstructed and allows us to greatly improve the amount of signal we can get back from a 3D cell culture model. So in this slide here, we took 3D cell culture models around 250 microns in diameter that were labeled with Cytox green nuclear stain. And we went ahead and imaged those models before and after we cleared them with both wide field and also confocal microscopy. And what you see on the right here, for spheroids that are non-cleared, so these 3D cell culture models that are non-cleared, even with confocal microscopy, we're only characterizing the periphery of these models. When we go ahead and actually clear these models and image them with confocal, we can characterize all the cells within these models instead of just those in the periphery. This is very, very important because if we're trying to look at how a compound is interacting with a 3D cell culture model, if we're only looking at the exterior, that is where the compound is, that's where the cells are most proliferative, that's where the cells are most exposed to nutrients, most exposed to gradients, and also most exposed to the compound. So that area is going to be very different from the interior of the 3D cell culture model, and especially so if we're looking at any kind of large molecule that has a challenging time with penetrating into these 3D cell culture models. So clearing here allows us to characterize about 3 to 4 times more cells in both wide field as well as confocal microscopy, and allows us to look at the entire population. So on this slide here, we're just showing this graphically. So as we go up through the 3D cell culture model and we get to about the mid-plane of 120 microns or so, what we're seeing is that with non-cleared, we're only characterizing maybe 10%, maybe 15% of the cells at that midline. But with the clearing, we're actually characterizing the majority of the cells that are actually in that model. So we're not biasing our results. Today, if you're trying to characterize a 3D cell culture model using an image-based assay, you're likely tremendously biasing your results and only characterizing the periphery, which is going to give you very inaccurate dose response curves, which I'll talk about in a moment here. Now, when we design a 3D cell culture assay, there's a lot of practical considerations that go into it. The first of which is always the type of model. We get a lot of questions from researchers about models and what types of models we have and which types of models are best for a specific question. And really this comes down to what the researcher is looking for. You can have a very simple model that is one cell type. You can have a model that is 3 cell types, primary cell culture. It really depends on what kind of research question you're trying to answer and what are your desired endpoints and what's the desired accuracy of the model. Do we have to have an incredibly high in vivo relevancy, or is a very simple HepG2 liver model adequate for your research question? That's going to drive the price, the throughput of this assay dramatically. The next part is identifying the minimum amount of data required for a research question. I think this point gets lost on a lot of folks, is that we don't want to acquire tons of data just for the sake of acquiring data. More data is not always better. In this context, the more data we acquire dramatically reduces our throughput. So for example, if we're doing a 4x imaging run versus a 40x imaging run in Confocal, the amount of data we're generating is going to be off orders of magnitude, 1,000 times more data with one compared to the other, and that data has to be processed. The acquisition time for acquiring that data is going to be much longer, and ultimately this causes us to generate more data than potentially we need. So we're always very focused on working with our clients to determine what is the minimum amount of data we need to actually answer the research question. If we need 4x and we're doing 40x, we're spending way too much time and money on the assay, whereas we can get the exact same answer for our research question with a much more inexpensive assay. Labeling and analysis. We focus a lot on what types of labels to use for a research question. This usually goes back and forth with the researchers. We try and optimize the labeling to get uniform penetration of antibodies throughout the model. And typically we use 3 to 4 labels depending upon the research question that we're going after, and are heavily dependent upon antibodies and getting antibodies that penetrate into 3D cell culture models. And then lastly, once we generate this huge amount of data, we focus with our clients on figuring out how to distill that down to an actionable insight. Pretty pictures don't really answer questions. So it's always about taking large amounts of data, terabytes of imaging data, and distilling it down to a singular report that a client can actually use to answer the research question. So we focus on this whole spectrum of questions before we even start an assay. It's very, very easy to go down the path of acquiring too much data or acquiring data that doesn't actually answer your research question. For labeling cleared tissues, so 3D cell culture models that have been cleared, typically we label tissues prior to clearing. Our technique, Visikol HISTO-M, is compatible with fluorescent proteins, immunolabeling, FISH, fluorescent proteins again. Fluorescent probes, sorry, the top here, but fluorescent proteins are compatible. A lot of tissue clearing techniques will quench fluorescent proteins, but we've developed our approach to actually be compatible with fluorescent protein and immunolabeling. And we've developed a number of techniques to get antibodies into very dense tissues. We've noticed a lot of issues with getting antibody labels into neuronal spheroids, which are quite popular these days, and have optimized protocols for all different types of 3D cell culture models, depending upon what specifically was required to get into those tissues and allow for uniform penetration. A major consideration in imaging 3D cell culture models is LED or laser excitation. This is a new trend that we're starting to see, is instruments going back to lasers. A lot of instruments initially had lasers. They were complicated. They broke down a lot, and instrument manufacturers switched back to LEDs. And then recently we see manufacturers switching back to lasers as simpler and more inexpensive systems are coming out to run those high content confocal and non-confocal systems. So laser excitation is really important for imaging 3D cell culture models. And the reason for that is this coherent light source allows us to get much better penetration of photons into our 3D cell culture model, especially if you're using any kind of U-bottom plate, like with the Corning ultra-low attachment plates. And instruments like the Opera Phoenix, the GE Incel 6000 and 6500, and the CX7 LZR, they have laser excitation, which allows much, much better imaging than you get on an LED system where you have a non-coherent light source. So we're comparing on the right here, side by side, the CX7 LED, which is a CX7 system from Thermo Fisher, and the CX7 LZR, the exact same system with an LED versus a laser-based excitation source. And we see a dramatic improvement in image quality by using a laser-based system compared to an LED-based system. We use our laser-based system for not only 3D cell culture models, but actually also imaging 1 millimeter and 2 millimeter thick pieces of mouse brain tissue. So laser allows you to just get much better penetration of photons in the tissue and a much better quality of image. And one of the things you'll see on LED-based systems is what we call foreshadowing. You'll definitely see this in a U-bottom plate, is you'll get a shadow of excitation below your 3D cell culture model, kind of like a halo. And this artifact is challenging to remove from your imaging run, creates a lot of problems for image analysis. But laser-based systems don't have that issue, and you just get much, much crisper images from a laser-based system. The next consideration we hear about a lot is the type of objective that you're using to characterize a 3D cell culture model. Most of the time, we're using 10 or 20x objectives. These are air objectives, non-water dipping objectives to image 3D cell culture models. We can get all the way to a millimeter, if not deeper, with an air objective on our system with tissue clearing. So there's some imagers that have immersion systems, water immersion or oil immersion systems, and those will certainly improve your image quality and your imaging depth. But with the use of tissue clearing, that improvement is far less significant. You're actually able to get much better images with tissue clearing than you would with a water immersion or an oil immersion objective on its own. So we see that it's really a myth. You don't need an expensive water dipping objective to image a 3D cell culture model. You can get by just fine with an air objective, very standard air objectives with using tissue clearing. Now, the next consideration is confocal versus widefield, the type of imaging system to use. We do a lot of assays with both. It really depends on the research question. If you need spatial information from a 3D cell culture model, you're going to want to go with confocal microscopy. But in many cases, you don't need spatial information. You don't need stacks of images from 3D cell culture models. You just need to acquire a high-quality image. In that case, wide-field imaging is adequate to answer your research question. It's much higher throughput. You don't get any depth information, but it's much higher throughput. The assays will be more inexpensive, and the data densities will be much lower, so much easier to process for a researcher on a per-model basis. The next thing we hear a lot about is if we're using confocal imaging, do we process Z projections or do we process stacks? Really depends, again, on the research question. A Z projection, though, just for some background, is when you acquire multiple sections, Z slices from a tissue, and you compress them all into one singular image and process that image. That can be useful for some applications, but the problem that we see is Z projections, they lose the spatial information that's inherent in these Z slices. You're losing that Z coordinate by compressing all those slides, all those images into one image. And one of the other things that we see when researchers do this is part of the reason a lot of researchers don't realize the bias in their assay is when you compress all these images together, you don't actually see that you're only acquiring imaging data from the periphery of a model. You think you're acquiring all the data, but in reality, you've only characterized the cells on the periphery of the model, and that's what your characterization is in a Z projection. So again, it depends what your research question is. We do a lot of work with Z-stacks, so processing data as three-dimensional spatial data sets and using that to answer complex questions like antibody penetration, for example, into a 3D cell culture model. Now, to dive into some examples of 3D cell culture models and how they're used, I want to start off with a first case study on personalized medicine. A question we commonly get, again, is why 3D cell culture models? One of the things that 3D cell culture models do is they mimic the complex microenvironment within tissues better than traditional 2D cell culture models. And in this case, that impact is very significant. So we've taken a wood cell line, a breast cancer cell line from Solaria. And in this case, we took the exact same cell line, we grew it in 2D and we grew it in 3D. And what we see is within the body, this type of tumor will express ER. When we grow it in a 3D cell culture model, it expresses ER just like it would within the body. We grow it in a 2D cell culture model, it does not express ER. This is highly significant because if we ran an assay with this cell line and we were looking for this specific type of ER expression, we wouldn't see it in the 2D cell culture model. So therapeutics that might work within the body, we're actually going to screen out because we're not getting proper expression of ER. So we get a much better expression of proteins that we're expecting in a lot of cases in 3D compared to 2D. And you'll see this on this slide here, is that when we have this wood cell line grown in 2D, which is in blue here, and we have it grown in 3D, which is in red, we see for fulvestrant, which is an ER targeting drug, that fulvestrant works very well with 3D cell culture models, but it doesn't really do anything with a 2D cell culture model. We're looking at cell proliferation here, which is the marker Ki-67. So in this case, what we're seeing is that this drug, we would not have seen its efficacy with doing a 2D cell culture assay. We might have excluded it from our screen with a 2D cell culture assay. Whereas with a 3D cell culture assay, we're seeing it work quite well. And that allows us to see that we're getting much better expression, and we're getting a much better mimic of the complex environment within the body in 3D compared to a 2D assay. Now, the same is shown here with tamoxifen. Tamoxifen reduces viability in 2D, but not in 3D. So really what we're trying to stress here is that it's very important, depending upon the research question, to mimic the complex environment within tissues. And if we don't do that adequately, we can actually get an answer to a question which isn't indicative of what's going to happen within the body. The next case study here is a very simple assay. It's a live dead assay. We've developed a whole bunch of assays that leverage tissue clearing with fluorescent labeling, with imaging. In this case, we took Hep G2 spheroids grown in Corning ultra-low attachment plates. We went ahead and stained these with DAPI and combined that with a kit from Thermo Fisher for live dead. And then we looked at what happened after seeding at different densities. So we seeded the Hep G2 cells between 500 and 16,000 cells. And we looked at how this changed the necrotic core over time. So we're seeing here is with a much higher seeding density, we're getting a much more significant necrotic core. So dead cells within the center of these models is shown in green here. And the reason I wanted to show this is a pretty unexciting application, but it's interesting because a lot of researchers until recently actually thought the center of 3D cell culture models was hollow. There was nothing there, it was just absent. And what we've shown is that there are in fact cells there. The reason why a lot of researchers didn't think there were cells there or organoids, 3D cell culture models were hollow is they simply couldn't actually characterize the interior because light was attenuating. As we're imaging these models, light's attenuating past 1 or 3 cell layers, and because of that, we're just not getting signal back. So a lot of these researchers who thought they couldn't characterize the interior of these models, or there was nothing there, it was really that they just weren't getting the data back from the interior, but there were in fact cells there. And a lot of researchers also think that it was impossible to label the center of 3D cell culture models and actually get labels into these models. And again, the problem was imaging, getting photons back. It wasn't actually labeling. We realized this early on after our experience with imaging whole mouse brains. We realized the problem couldn't be labels. It had to actually be imaging. And that's exactly what's going on here. We're showing the ability to look at all the cells in the population and to characterize the necrotic core of these models. Now, the last case study I was going to focus on here was looking at Hep G2 cells and cell proliferation. So very, very common assay, a lot of researchers do. So we're looking at Ki-67 antibody labeling. And in this case here, we're looking at Taxol. So we're using Ki-67. We're clearing the tissues with Visikol HISTO-M, and we're imaging. In this case, we're imaging with Visikol HISTO-M with a confocal microscope and generating dose response curves. So here we're looking at the comparison between tissues that have been cleared and tissues that have not been cleared. And what we see when looking at Ki-67 for cell proliferation, there is a substantial difference between clearing the model and not clearing the model. The reason why this is so important is because this shows that with characterizing the entire population of cells, the results you get are far different than if you're only characterizing the periphery of cells, which is what most researchers are doing today. So if we're looking at only the exterior, we're going to think cell proliferation is much higher than it actually is because we're characterizing the cells that are most proliferative on the outside of these models. And to show this a little bit further here, if we break down the 3D cell culture model into inner, middle, and outer, we can start to see the Ki-67 % positive cells in each one of these different parts of the 3D cell culture model. And we see there's a very, very different response depending upon where we are within the 3D cell culture model. This is very important for looking at large molecules like antibodies, where we're going to have a very different response between the exterior and the interior. And if we're only biasing our results towards the exterior, the assay is going to show a very different result than what's actually happening and what's probably going to happen within the body. So as a company, we focus a lot on 3D cell culture models and trying to figure out how to best extract quantitative 3-dimensional information from these models. And again, it really depends on the type of question that you're trying to answer and the type of model you're trying to work with. But again, by looking through those different parameters on how best to develop your assay, how to minimize the amount of data that you want to acquire, you can develop a 3D cell culture assay that does both improve your ability to predict what's going to happen within the body, while also reducing costs and allowing your program to move forward more quickly. So thank you so much for the opportunity to present. And if you have any questions at all, please feel free to email me at michael.johnson@visikol.com. Thank you.
Thank you, Dr. Johnson, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. Our speaker will follow up with your questions via email. All right, let's get started. Our first question is, what is the most challenging component of executing 3D cell culture assays?
I would say probably the most difficult part we see from researchers, again, this depends on the type of assay they're doing, but if they're doing any kind of imaging-based assay, we see the most challenging part is actually the data processing, turning a bunch of really nice images into something useful. We see this is where most researchers struggle, especially pharma companies. Pharma companies have a really great expertise in what we see as the biology, a lot of times the imaging as well, but when they get to generating terabytes of data per day, actually distilling that down and answering their specific research question or optimizing some type of algorithm to do that, we see a lot of problems there, a lot of challenges. So that's where we've seen probably the most problems for researchers. We've actually recently developed software to address that. So if folks are interested in learning more about that, shoot me an email. I'm actually looking for beta testers to test out the free software. But doing that and answering those questions and taking really pretty pictures and turning them into a table or turning them into a report, that's where we see the gap for most researchers. It's a lot of data to process, and it's an expertise that a lot of life scientists just don't have a background in computer vision or image processing.
All right, and how do you choose the right model for a 3D cell culture assay?
This is a common question that we get is, what models do you guys have to run assays? And the way that we look at this is we're pretty agnostic on the model. We always ask the client, well, what is your specific throughput, cost, and validation criteria? Because you could run a very simple assay, or you could run a very complex assay with a really complex model where you have a lot of validation criteria. You've done a lot of assays to validate your model's actually performing the way that it should. But for many researchers, they don't need that. So I think it really depends on the researcher. We work very closely with researchers to try and educate them on this, that there isn't a liver model. There are a varying spectrum of liver models, and the one that's right for you really just depends. I think a lot of companies are trying to move towards, like we see microtissues and we see organoids, but there's very unlikely to be a small, tiny liver in the dish within the next few years. It's really about finding that liver in a dish-like thing that answers your research question and also meets your cost and your throughput criteria. So I would say it's really just working through those questions and understanding the limitations. These are not yet microorgans on a plate.
All right, we have time for one more question. How do you analyze your data to characterize your model?
So this, again, depends on like the type of question that we're trying to answer. But to give you probably the most complex answer or complex assay we do is if we're doing confocal imaging, we're generating Z-stacks of images from a 3D cell culture model, what we typically do is we use our imager to generate TIFF files, so just generating lots of image files. And then from those image files, we've generated a whole wide range of algorithms to answer research questions that we commonly do for our clients. So for example, probably one of the most common ones that we get is co-localization. So we want to count the number of nuclei that express, for example, or number of cells that express, let's just say, DAPI and Ki-67, and count the percent of positive Ki-67 cells. So in that case, we're looking at the two different channels, we're putting those on top of one another, and we're seeing where that co-localization occurs and counting the percent of cells that express both of those markers. It's a very common question that we get, but all of that processing, all that analysis is done actually externally in software that we build. We've just seen a lot of challenges with doing 3D image analysis and how to build out a lot of software internally that we use with our clients.
All right, I would like to once again thank Dr. Johnson for his presentation. And I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December of 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.